Right. So, starting with our new topic, that's biodiversity and conservation of species. So, when we go ahead and talk about biodiversity, we did discuss about biodiversity when we did the topic of classification, and we talked about biodiversity being the variety of living organism and the genetic variation that can be seen within a habitat, right? Now, when we talk about the actual way of defining or the actual way of actually discussing what biodiversity is, again, the exact way of defining biodiversity is difficult, right? Just like when we talked about species, we talked about the different species model, we talked about the different ways of defining the term species, and we talked about the limitations associated with it. Similarly, when we talk about our biodiversity, coming up with a definition for biodiversity is not easy. Again, their number of different species is a useful basic way of defining it, but technically speaking, it's a lot more complicated, right? So when we talk about biodiversity, biodiversity can be assessed on different scales, right? We can start from talking about at a level of species. We can talk about the variation between species, or we can talk about genetic variation. So it's not just that you're just saying it's a number of different living organisms. You can start it at the number of different living organisms, and then you can go down different scales. You could go down to the smallest possible level, that is the genetic level. When we go ahead and talk about it, so this is the definition that is generally used: variety of life on earth, from the smallest microbes to the largest animals and plants. So when we go ahead and talk about it, when you're talking about biodiversity, are you just talking about the different number of species? What about when inside a species there is genetic variation between the species? Is that going to be included? Right? What about the variety of ecosystem? What about the variety of habitat? What about the variety of your populations? What about the variety of your communities, right? So when you go ahead and talk about it, biodiversity can be seen at different scales, which is why defining biodiversity is something that is not easy. And just like when we talked about our topic of species, so we talked about different models. Just like that, we can assess biodiversity, or we can study biodiversity at different levels. One of the ways we can study biodiversity is by looking at the species, right? What is the variation between species? Now, how do we measure this variation, right? How do we find out what is the variation in species? What is the biodiversity of our species? One way that you can do this is by measuring the species richness of a particular area, right? That is the number of different species in an area. So you take an area and you calculate the number of different species. That will give you species richness of that particular area. The second thing you can look at is relative species abundance. What is relative species abundance? The distribution of different species. So, for example, how evenly are they distributed, or how oddly are they distributed? Are they spread out throughout commonly, like are they spread throughout equally, or are they spread out in small groups? Are they clumped together in just one area? Right? What is their distribution? How are they spread out throughout throughout the area? What is the distribution of different species? Right? That is going to be relative species abundance. So these are the two ways that you can go ahead and you can actually measure what? You can measure biodiversity at the level of species. Now, when we go ahead and talk about our species richness, so biodiversity varies enormously around the world in terms of number of species. If we talk about the wet tropical area, right? Wet tropical area are the areas where you would have the highest biodiversity, like the Amazon forest, right? Almost every tree, for example, you will see would be a different species. Then inside a tree, imagine the different number of animals, different number of insects, different number of, your microbes that are living in that particular one tree, right? So you can see that the species richness would be extremely high in wet tropical areas. As we move away from the wet tropical areas, the species diversity tends to increase, right? And when you go ahead and so that is what happens in terms of species richness. Then we have to go ahead and discuss a term known as endemism. What is endemism? Okay, what is endemism? Endemism is another important way of measuring biodiversity. This is another way that you can measure biodiversity. So one of the ways is species richness, right? The other way you can use is endemism. What is endemism? By looking at the number of endemic species in an area, right? When a species evolves in geographical isolation. What do we mean by geographical isolation? So there's certain species, for example, they're growing on an island. So they're geographically isolated, meaning that because of geography, because they're not able to travel from one area to another, they are isolated. So geographically isolated animals, when you're going to talk about it, they can only be found in one place, that particular island. So they will said to be endemic to that place. For example, let me give you a big example, Australia, right? There's so many animals that are endemic to Australia, like your kangaroos, right? Like your koala bears. All of these, your marsupial animals, marsupial animals that have a pouch. So these animals, they are generally endemic to Australia, and they are generally found in Australia because of geographical isolation. So endemism is another way of measuring biodiversity, right? So when you go ahead and talk about it, when you go ahead and talk about it, just like that, there are lots of different animals that are unique to certain areas or that are unique to certain countries, right? So, but when we talk about it, the areas with the greatest biodiversity are not always the same as the areas with greatest number of endemic species. Remember that. So, for example, that rainforest, like I told you, Amazon rainforest has one of the highest biodiversities. But that does not mean that it has the highest number of endemic species. Species found in Amazon rainforest perhaps could be found in other places as well. So when you go ahead and talk about it, this is why it is so difficult to prioritize the areas for conservation of species. When we're talking about conservation, how are we going to prioritize which area we're going to go for, which area are we going to try to conserve? Because again, biodiversity is not the same thing as endemism. Endemism can be used to measure biodiversity, but just because one area has a lot of biodiversity does not mean it has a lot of endemic species. So when we go ahead and talk about it, there are a few rules when you go ahead and talk about it related to your biodiversity, right? So the current model that we use, what is the current model that we use to discuss biodiversity, or what are the current things we look at when we are trying to find out whether an area has rich biodiversity or they do not? What is it? First of all, it has to be a very stable ecosystem. A stable ecosystem allows many complex relationships to develop between species, right? That's the first thing. A very stable ecosystem has to be there for there to be high biodiversity, because the more stable the ecosystem, the more complex the relationships can develop. And as a result, there can be more biodiversity. There needs to be a high level of productivity. Why? There needs to be high level of producers. Greater the number of producers, greater the variation in the number of producers, the higher levels or higher niches can be supported. Primary consumer, secondary consumer, greater the variety of niches or greater the number of niches that can be supported. And when organisms can grow and reproduce rapidly, it is more likely that mutations will occur. And if there are going to be more mutations, there are going to be more adaptations. If there are going to be more adaptations, there's going to be organisms are going to be better, have a better chance of survival on as a result, when they're going to have a better chance of survival, they will start, they will start taking on more roles in the ecosystem. They will start taking on more niches in the ecosystem. So again, biodiversity will increase. So when we go ahead and we talk about our this topic of endemism, right? So endemism, once again, it does not really give you an idea of what your biodiversity is, but it is one of the ways in which you can assess how much biodiversity is present. Now, another way when you're going to go ahead and talk about it of measuring your biodiversity is by calculating species abundance, right? The absolute number of species is not the only important factor in biodiversity. Relative species abundance is also significant. Now, I'm not going into details of relative species abundance. We're just discussing the quadrant method over here. There are other methods as well, but we are going to do that in our unit. So the way you can measure relative species abundance, one of the ways is you use a quadrant method. So we have a frame, and what we do is we take a random sampling area. After taking a random sampling area, you will take this frame. The frame is divided into multiple equal sized quadrants. You will place the frame on your sample area, and then you will count the number of different flowers. For example, you can see in each quadrant, right? So, for example, you can see different flowers, right? For example, this is a different plant. This is a different plant. Maybe this is a different plant. This is a different plant. So you will go ahead and you will count the number of different plants you can see in each quadrant. So you'll mark this quadrant number 1 and you will write down, oh, I found one, for example, dandelion, or I found one buttercup, whatever. So you're going to go ahead and do your note down the number of different plants that you can see in each quadrant, right? And then if you want, you can go ahead and you can turn it into percentage. So this is another way of measuring your biodiversity. Now, what about calculating our biodiversity, right? These were different techniques that we were using. But what about our actual calculation? So actual calculation can be done by using something known as the diversity index. What is our diversity index? Diversity index is equals to n bracket n minus 1 divided by sum of n bracket n minus 1. When we go ahead and talk about it, so our capital N stands for the total number of organisms of all species. The small n stands for total number of organisms of individual species minus the abundance of different species. For example, take a look. Let's go ahead and discuss this. So they have given you this table, okay? You have the different species over here. Fine. You have the number of individual species. So 4 of the dragonfly, 12 of the mosquito, 3 of the water boatman, 4 of the shrimp, 17 of the copepods, and then your total number of organisms are 14. Your formula is basically going to be D is equals to N bracket N minus 1 divided by sum of N minus 1. First thing you're going to do is you're going to make a table. So you're going to make a table and your next column is going to be n minus 1. 4 minus 1 is 3, 12 minus 1 is 11, 3 minus 1 is 2, 4 minus 1 is 3, so on and so forth. Fine. After that, you're going to make your next column, which is going to be n bracket n minus 1. So 4 multiplied by 3, 12 multiplied by 11, 3 multiplied by 2, so on and so forth. After you've made the table, you will have to add all of these up. So you're going to add all of them up and you're going to end up getting your final answer, right? So your formula is going to go like this. D is equals to N. Capital N is 40. So you write down 40. Bracket 40 minus 1. 40 minus 1 is 39. Divided by your sum of N bracket n minus 1. So sum of n bracket n minus 1 is 12 plus 132 plus 6 plus 12 plus 272. So what were we getting? We were getting our 12 plus 132 plus 6 plus 12 plus 270. So 40 multiplied by 39 is 1560. Divided by, if I add all of these up, my total is coming 434. If I divide them, my diversity index is going to come 3.6, right? And greater the value you're getting, greater your biodiversity index, the greater the biodiversity of that particular area. So this is how you're going to be doing calculations. Are the calculations part clear, Basil?
Yes.
Okay. Now, when we talk about biodiversity, how is it that biodiversity is changing, or how is it varying as we go across, for example, our area, right? So when we go ahead and talk about it, we talked about that high biodiversity is seen when you have a stable ecosystem, when you have high levels of productivity, and where organisms can grow and reproduce rapidly. These were the conditions that we had stated, that in these conditions you generally observe high biodiversity. So generally speaking, when you have an extreme environment like a desert, the biodiversity observed is going to be low, right? This type of an ecosystem is unstable and it is very susceptible to change, meaning that if in that same desert there's a sudden burst of rain, a sudden rainfall, that will cause a lot of different plants to start growing. As a result, a lot of different animals are going to have a better chance of survival. Their population is going to start increasing. And as a result, the ecosystem is going to change, right? So when you go ahead and talk about it, these sort of ecosystems or these stop of environments are really, you can say, unstable, right? When you go ahead and talk about it, these type of environment not only are they extremely unstable, but they also have some unfilled niches. What is niche? Role, right? So what can happen that any incoming organism or a new organism can quickly occupy that role. It can quickly occupy that niche. It can divide very rapidly, and it can overpower any existing species that is currently present. So that is why in extreme conditions, biodiversity is low, because the environment is unstable, because they are very susceptible to change, and they have a lot of unfilled niches. In a less hostile environment, biodiversity can be very high because the stable ecosystem is stable. And even if a new species moves in or out of the ecosystem, it does not really have an effect on the biodiversity or the ecosystem or the food web of that particular area. Biodiversity can be lost due to natural events such as volcanic eruption or flooding, and also as a result of human activities, right? Now, when we go ahead and talk about it, biodiversity is not constant. Fine. For example, your animal species migrate, right? So if migration is taking place, your biodiversity of an area is definitely going to be changing. Then biodiversity can also change depending upon the time of the day. For example, owls and bats are more visible during night because they're nocturnal animals. You might not observe them during the day. So biodiversity depends upon the climate, it depends upon the time of the day. And along with that, it also depends upon the temperature. So the picture of biodiversity in an area can change throughout the year, right? So when we go ahead and we talk about it, whenever we're measuring our biodiversity, we are going to, it's going to depend upon what time, what duration, what climate we are measuring it in. Okay, so remember that biodiversity is never constant. Just because you measured it in summer does not mean you will get the same results in winter. So biodiversity is changing constantly throughout. Now, what about biodiversity within a species? So we were talking about biodiversity between species. What about within a species? right? Biodiversity within an individual species is a very important concept. Why? Because of gene pool. The gene pool of a species is all of the genes in the genome, including all different variants of each gene. So if there are genetic variation, they are included in the gene pool. So all of the genes in a genome, including all the different variants, they're all going to be part of the gene pool. And when you go ahead and talk about it, there is something known as gene frequency and allele frequency. What is gene and allele frequency? So when you talk about mutations, you know that mutations are changes in the DNA structure. Some mutations have no effect on the phenotype, some mutations are beneficial, and some mutations are harmful. Mutations can increase the gene pool of a population by increasing the different number of alleles available. The relative frequency of a particular allele in a population is called the allele frequency. The relative frequency of a particular allele in a population. Relative frequency of a particular allele in a population is known as the allele frequency. If a mutation is advantageous, if it's beneficial, the allele will be selected for, it will be selected again and again and again. Why? Because it's giving your organism a better chance for survival, right? So that allele is going to be selected again and again and again, and the frequency of that allele in the entire population will increase. If the mutation is harmful, natural selection can remove it from the gene pool. Or even if it's not removed, you will see it at a lower frequency compared to the beneficial mutation. So when you go ahead and talk about it, do remember that a harmful gene or a disadvantageous gene can become an advantageous gene in a different condition, right? If I were to give you an example, what do I mean by that? Let's talk about sickle cell anemia, right? Sickle cell anemia, there are three possibilities. Either both the hemoglobins are normal, or one hemoglobin allele is normal and the other is for sickle cell disease. And the other option is that they are homozygous recessive, where both the alleles are for sickle cell anemia. Now, when we go ahead and talk about it, in the African population, this was basically observed that the people who had the sickle cell gene, they were resistant to malaria. Right? So what ended up happening was this, that because the people with normal hemoglobin would be susceptible to malaria, they would end up dying because of malaria, and the number of people that were homozygous dominant, they started to reduce. The number of people that were homozygous recessive, they also started to reduce because, of course, they had sickle cell disease and they would end up dying from the disease. Those people that were heterozygous, the frequency of those people started to increase in the population. Why? Because first of all, they had the dominant allele for normal hemoglobin, so they only had very mild symptoms. At the other hand, they did have the sickle cell gene, which allowed them to be resistant to malaria. So their population with heterozygous, they started to increase. So in this particular situation, what happened? That the natural selection led to reduction in the frequency of people that were homozygous dominant. So even though the sickle cell trait or the sickle cell allele was disadvantageous, but because of that particular environment, because of that particular condition, the sickle cell allele actually became beneficial. So just because one allele is harmful or disadvantageous does not mean it will always be disadvantageous. If the environment changes, it could possibly also end up becoming beneficial, right? So that is what happens. Now, what about now? How do we measure all this? How do we measure allele frequency? How do we measure this entire genetic variation, right? Genetic variation within a population is an important measure of biological health and well-being. Without variation, any population is going to be vulnerable. So when we go ahead and talk about it, scientists look at the proportion of population which is heterozygous for a given feature. Heterozygous have more than one allele for a given feature, right? You know that, for example, for hemoglobin, one was normal, one was sickle cell. So they have more than one allele for a given feature. So if you want to measure genetic variation, you take a look at the heterozygotes. As a result, when we are doing our calculations, we use heterozygosity index. We use the heterozygosity index to go ahead and calculate the genetic diversity of a population. The heterozygosity index is number of heterozygotes divided by number of individuals in the population. This is going to give you your heterozygosity index. And heterozygosity index, the bigger, the better. The higher proportion of heterozygotes that you have, the greater genetic diversity can be seen in that particular area. Any confusion? Anything you want to ask so far?
No, ma'am.
Okay. So when we go ahead and we talk about our entire topic, so this is your heterozygosity index that you need to know about. There is another, I think, yeah, the Hardy-Weinberg equation we have to do. Okay, we have to do that as well. Before we do, or should I do that first? Okay, let's just go ahead and do that first since we're going over it. Okay. When we talk about a population, what is a population? Population is a group of individuals of the same species occupying a particular habitat or a particular niche within that habitat, right? A group of individuals of the same species occupying a particular habitat or a particular niche within that habitat. Now, when we go ahead and talk about it, what is a habitat? Habitat is the place where it lives, including both the physical elements and the biological elements. Niche of an organism is its place within the ecosystem, right? Including its habitat and its effect on other organisms. You don't have to go into details of niche and stuff. That's again in unit 4. Now, when we go ahead and we talk about the genetics of a population, so first of all, we were talking about genetics of a species, right? Now we're going to go ahead and talk about our population. So a population is a group of same species. So in a population genetics, we take the gene as a unit of evolution and look at how the genetic composition changed over a period of time. The sum total of all alleles in a population at a given time is called the gene pool. And when we go ahead and talk about it, so the frequency of alleles in a population is not fixed. So population genetics is the sum of total of all alleles in a population at a given time. That is the gene pool of population. Gene pool of species was what? The number of different genes that can be found within the species, right? This is the number alleles is also going to change because of natural selection, because of different adaptations, and we will study the adaptations as well. When you go ahead and talk about it, so generally there is a selection pressure. That selection pressure causes the selection of that particular allele that gives the organism a better chance for survival. For example, there is a chemical known as warfarin. Fine. Warfarin is basically, it's an anti-clotting drug. Now, generally, it was used in rat poisons, right? So it would be put in a rat poison. The rat would eat it, their body would stop clotting, and they would die from internal bleeding. Now, when you go ahead and talk about it, there were certain rats that developed a mutation against warfarin. Now, what happened? The warfarin acted as a selection pressure. Those rats that had developed the mutation that allowed them to resist the effect of warfarin, they started to thrive and they started to increase in number. And as they started to increase in number, they started to pass on the mutated gene to their offspring, and then to the next generation, and then to the next generation. And as a result, the frequency of the mutated recessive allele started to increase, right? So selection pressure is seen when a change occurs in the environment. So when you go ahead and you're going to talk about it. So in this particular situation, the warfarin acted as a selection pressure, right? So the rats that have the mutated gene, they had an advantage in these new conditions. Fine. Rats with the alleles that give resistance to warfarin were more likely to survive, they were more likely to reproduce, and they were more likely to pass on the resistance gene, right? Similarly, the same warfarin acted as a negative selection pressure on those rats that did not have the mutated gene. So those that had the mutated gene, for them, it was sort of a positive pressure. On those that did not have the mutated gene, it was a negative selection pressure, right? So what happened? That those that did not have the mutated gene, they were more likely to die. They were more likely to not reproduce. And as a result, the frequency of the susceptible allele will be decreased. Eventually, majority of the rats became resistant to warfarin, and we had to come up with different types of poisons. So when you go ahead and talk about it, speciation involves a change in the allele frequency within a population, and it is driven by selection pressure over a period of time. So that is why when you talk about your allele frequency is not fixed in a population. It is going to be varying. It is going to be changing over a period of time. Which is why we come to this concept. Fine. So the number of individuals carrying a certain allele in a population determines the allele frequency. What are the number of individuals carrying a certain allele, right? So the number of individuals carrying a certain allele in the total population will determine the allele frequency. So it will tell you what proportion of the individual carries a certain allele, and we usually express it as your as the total of 1, right? So we take the total as 1. So when you're going to go ahead and talk about it, what proportion of the individuals are carrying a certain allele, and we describe it as 1, right? Then we go ahead and we use this general formula. The general formula is this, that we take the allele frequency for the dominant, and we take the allele frequency of recessive phenotype, and when we add them up, they should be equal to 1. So the frequency of the dominant allele plus the frequency of the recessive allele is going to be equal to 1. But there's a slight problem with this equation. So we are talking about how to calculate allele frequency right now, right? We're talking about the calculations for allele frequency. So this is one of the ways that you can find out what is the number of homozygous, what is the number of your recessive allele. But when we go ahead and talk about it, this is not a good method. Why? Because how are you going to distinguish between homozygotes and heterozygotes based on phenotype? Because this is based on phenotype. You are differentiating dominant and recessive based on phenotype. So you're counting the number that are dominant, counting the number that are recessive phenotype, and you're saying that adding them up is equal to 1. But how will you know how many of them are heterozygotes? How will you know how many of them are homozygotes? Because both heterozygotes and homozygotes will give you the dominant phenotype. This is where the Harding-Weinberg equation comes into play. This is the equation that you can go ahead and use to actually do your calculations and take into account your heterozygotes as well, right? The amount of change that takes place in the frequency of alleles in a population indicates whether the population is stable or it is evolving, right? If the change in the frequency of allele is happening, right? If the change in frequency of allele is happening, the frequency of allele is changing, that would give you the idea that the population is still changing or the population is still evolving. It's not yet done evolving. On the other hand, what if your allele frequency is remaining the same? If allele frequency is remaining constant over a period of time, that is basically giving us the situation that now this population is stable. This population is stable and there are no other evolutionary influences. There are no evolutionary influences. There are no evolution taking place. So in this situation where your population is stable, we can use the Hardy-Weinberg equilibrium. We can use the Hardy-Weinberg equation. This is for when our population is stable, when it is not changing. Fine. If it is changing, then we can't use this equation. So the equation is this. When we go ahead and talk about it, the equation is this, right? So the equation is your Hardy-Weinberg theory basically states that in a population that is not evolving, the allele frequency will remain stable. If the population is evolving, the allele frequency will change from generation to generation. So when you go ahead and talk about it, this is the equation. Frequency of homozygous dominant genotype in a population, and you square it, plus 2pq, which is the frequency of heterozygotes genotype in a population, plus q square. Q square represents the frequency of homozygous recessive genotype in a population. And all of that is going to come up to be equal to 1. For example, people who inherit the allele for dominant pigmented trait may have the genotype AA or capital A, small a, right? They can be homozygous dominant or they can be heterozygous. People who are albino are homozygous for the allele, that is, they are homozygous recessive. Now, test on a sample of North Americans showed that the frequency of albinos in the population was 1:20,000. So 1:20,000 is what? The recessive allele, right? 1:20,000 is the recessive allele. So that's our Q square. So 1/20,000 is equals to Q square. So to calculate Q, you will do an under root. So Q is going to end up coming as 0.007. Now, remember how we said P plus Q is equals to 1. So P is equals to 1 minus Q. P is equals to 1 minus 0.007. P is equals to 0.993. Now, once again, we need to know the heterozygotes. We don't need to know the how many of them are dominant, how many of them are recessive. We need to know the heterozygotes, because greater the frequency of heterozygotes, the greater the biodiversity that you're observing, right? So when we go ahead and take a look, now you will use the Hardy-Weinberg equation. Fine. Now, when you're going to go ahead and take a look, what are you going to do? You are going to start with P square, 0.993, square it, plus 2, 0.993, and then you will write down Q, 0.007, plus Q square. Q square was 0.007 square. You will see whether the answer is coming up to 1 or not, right? So when you go ahead and talk about if the answer is coming up to 1, what does that tell you? That first of all, this is a stable environment, right? It is a stable environment. If the answer is coming up to 1, you know that it is a stable environment, right? You know that this is a stable environment. And when we go ahead and we talk about it, so P square was what? The number of homozygous dominant species, right? So 0.9933 square is going to give you the number of homozygous dominant. Q square is going to give you 0.007 square. That's going to give you the number of homozygous recessive. And 2PQ is going to give you the heterozygous genotype. So that's how you can use your equation, Weinberg equation, to calculate the dominant, heterozygous, as well as the recessive genotype allele frequency in a population. And if the total is coming up to 1, that is another way of confirming that your species is indeed at a stable state and it is not evolving. Is this all making sense?
Yes.
Okay, so what are the conditions in which we can actually apply our Hardy-Weinberg equation? So there's certain conditions, right? Like I told you that your population must be in equilibrium. Your allele frequency needs to remain constant over a period of time. So what are the conditions? There should be no mutations taking place. Fine. there is random mating taking place, meaning that the selection of mate is going to be random. The population is going to be large. The population is isolated, meaning that either it's genetically isolated or geographically isolated, and basically it's geographically isolated. There is no immigration taking place, no new species is moving in, no species is moving out, and there's no selection pressure, right? In these conditions, you can go ahead and apply the Hardy-Weinberg theory. But of course, if we talk about our real world, it's not possible for any population to meet all of these conditions. As you know, a population is constantly changing. There will be some selection pressure. The population is going to end up developing some mutations. When you talk about isolation of a population, that can also go ahead and change. So as a result, the allele frequency over a period of time is generally always changing, right? And when we talk about upsetting this gene equilibrium, gene pool equilibrium, upsetting the gene pool equilibrium is what results in the formation of a new species. Now, when we talk, so discussing all of these, now let's discuss these conditions in more detail. When we talk about our mutations, so for the allele frequency to remain stable in a population, no mutations must occur, because mutation is what? Change in the genetic material. So if there is a change in the genetic material, the alleles have changed, right? Now, as you and I know that it's not possible, because spontaneous mutations tend to occur generally over a period of time. Spontaneous mutations occur all the time within a population. As we go ahead and talk about it. So when we talk about our, so that is, when you talk about it, there can be two types of mutations. You can have mutations in the somatic cells, or you can have mutations in the germline cells. Germline cells are the cells that create your sperm and your eggs, right? If there is a mutation in the somatic cells of animals, they will not be passed on to the offspring, and they may not affect the individual themselves. For example, that mutation may have an effect or it may not have an effect. We have discussed this. But if the mutation is in the germ line, that means it's affecting the egg in the sperm, it will affect the alleles of the next generation, right? Then when we go ahead and talk about it, although mutations do occur continuously, they do not affect the population very rapidly. Fine. Occasionally, mutations arise that will bring benefit to an individual, and they will slowly become established within the gene pool. Now, the second condition is that there is random mating. Now, in a normal population, we have non-random mating. One of the most important requirements for a gene pool to remain in equilibrium is for random mating to take place. Random mating takes place that the likelihood of any two individuals in a population mating is independent of their genetic makeup. So it does not depend upon the genetic makeup. The likelihood of two individuals mating is going to be the same. If mating occurs randomly, the frequency of the population will remain the same. The frequency of alleles in the population will remain the same. But generally, in reality, we see non-random mating. Non-random mating occurs when some features of the phenotype affects the probability of two organisms mating, right? And as a result, natural selection is going to take place. What do I mean? Think about a peacock. A peacock, greater the tail of the peacock, the more impressive the tail of the peacock, the better chance of that peacock has with mating as compared to the peacock that perhaps has a smaller tail, right? So it depends upon the phenotype. It depends, the mating depends upon the phenotype, and that also causes natural selection to take place, because the peacock with a bigger tail is going to have a greater chance of mating. There's going to be greater chance of that genotype passing on, and natural selection can take place. A deer with very large antlers, right? Greater the size of the antlers, greater the size of the antlers, the more likely they are to attract the other species, and greater chance of mating taking place. This is non-random mating, right? And that is the same thing that you see in human population. Then, when you talk about population is large, that is not true. This theory is only applicable if the population is large. But as we know, so basically, why does it have to be large? Because maintaining genetic equilibrium depends upon random assortment of alleles, right? Large populations containing many individuals usually have large gene pools, and the chance of losing an allele by a random event is reduced. Fine. So because it depends upon random assortment. So large population, large gene pool. So less chance that any gene will be lost because of your random assortment. So when you go ahead and talk about it, let's say that there is, a small population. In a small population, if there is a percentage of animals that have the required allele or have a certain allele, what happens if that group or that individual is lost because of a disease, because they were eaten by the predator? The allele is lost, even if it is beneficial, even if it allows for adaptation to take place, that particular allele is lost. So you need to have a large population. But as you know, that this is not happen. It does not really happen in real world. In real world, we have got populations of different sizes. Then the other requirement is that the species should be isolated. There should be no migration of organisms. There should be no species coming in and coming out. But that's not true. Pollination is always taking place. Insects are carrying pollen from one area to another. Wind can carry pollens for miles. Male animals can, frequently leave their families and they can go to different areas. They can go to different other populations. Animals can migrate because of weather. Animal can migrate because there's not enough, food available in one area, or there's lack of resources, so they may migrate to another area, right? So when you talk about, for example, those that's reproduced by spores like your fungi, so again, those spores are able to travel a great distance, carried by water, carried by wind. So when you talk about it, there's always an exchange of gene flow occurring, right? There's always a gene flow occurring. So what happens that this tends to make different populations more similar, but it constantly changes the allele frequency. So again, this condition is there. The last condition is that there should be no selection pressure for this equilibrium to apply, right? So all alleles should have the same level of reproductive advantage or disadvantage, right? There should be no allele that has a greater advantage, because that is what happens in selection pressure. That someone has an allele that is advantageous, so that allele gets selected for. But for the Hardy-Weinberg equilibrium, all of the alleles should have the same level of advantage and disadvantage. But as you and I know, that is not the case in real life. Many alleles are neutral in their effect, right? But some alleles do end up coding for a phenotype which gives an advantage or a disadvantage to an individual. So that is what we know about that natural selection is going to end up taking place, right? If the environmental condition changes, if there's a new selection pressure, again, you're going to end up seeing a result in the change in conditions. Some alleles, which are neutral, might end up becoming advantages, or they might end up becoming disadvantages, right? So as a result, the genetic makeup of the population will be changing. Natural selection is going to take place. Selection pressure will drive speciation as the environment is going to change, and you're going to end up observing the change in allele frequency. Uh, so again, that is what happens in real life. The requirement for Hardy-Weinberg equilibrium is, of course, that there should be no selection pressure. Any confusion so far? Anything you want to ask?
No, ma'am.
Okay. Okay, Basit, we'll go ahead and stop it over here for today. So we're left with, I think we're still left with a good amount of topic. We have to go over our speciation, we have to go over our adaptations, and we have to go over our conservation of species. So we'll stop over here for today. And in the next class, we'll go ahead and we will then continue on with our topic. All right.
Okay, ma'am. Thank you.
Okay. Thank you so much, Basit. I'll see you in the next class. Have a good day.